And although Ron was not slated to be part of the final panel, if you have questions specific to the NETRF, please go ahead and write them down, and we'll try to address them, and we'll invite Ron up to the podium at that point. So so Pam asked me to talk about genetics, and I can spell DNA, and that's about where my expertise ends. So I decided to sort of reframe the discussion and call it Deciphering Cancer Genomics, Promises and Pitfalls. And so that's a warning that although there's plenty of promise, there are a number of pitfalls too. So let's kind of go through that a little bit. So the concept is if only we had the blueprint of what makes the cell tick, what makes the cancer cell tick, we'd all of a sudden have a way of predicting cancer in people who don't have cancer. We would have a way of preventing cancer in people at high risk of cancer. We'd have a way of diagnosing cancer earlier. We'd have a way of treating cancer better, and we'd have a step towards a cure for cancer. It sounded so simple back 20 years ago. We just need to know what the gene is that's causing the cancer. And here's a pill filled with DNA that's intended to do exactly that. Well, unfortunately, there are more pitfalls than promises. DNA is a dynamic blueprint. It's not a set guide. It actually changes over time, the tumor cell DNA, that is. And so since the tumor cell DNA is really can change over time, it can change in our Darwinian way and evolve. And it can evolve to survive. And if the tumor cell evolves to survive, that means the tumor cells can change as they go through in time. And we're just starting to learn that for other cancers, and we're a little bit behind in neuroendocrine tumors, but there are a number of projects, I think, where we can catch up. The DNA alterations come in many shapes and forms. It's not just a simple mutation. What does a mutation mean? A lot of mutations are not really deleterious. They don't actually have any downside. Some mutations are huge and the cell just dies. So there are many different types of mutations. Some of them are readily tested for, others a little bit more complicated to test for. I think that one of the that many people don't appreciate that when we talk about sequencing the genes in the DNA in a tumor cell or in a normal cell, that's actually only about 10% of all the DNA. 90% of the DNA doesn't eventually get expressed as proteins. So we used to think of all this DNA in between the genes as kind of junk DNA, but Mother Nature doesn't make junk. Mother Nature's smarter than that. So that DNA in between the genes, we now know is actually important. And that DNA in between the genes can influence which genes get turned on and which genes get turned off without an actual damage to that gene. So it's much deeper than just looking at the genes and understanding what the changes are. The sequences in between the DNA may have some of the secrets that we still have to discover. What's more is we can identify some genetic changes and say, well, that's an important genetic change, but we don't have a drug for it. So it's nice to know that you have a gene that we can recognize as an important gene, but if we don't have a drug for it, we're not going to help you. So, so there's a lot of effort to try to develop drugs for some of these other genes that we're finding. I think that understanding the genetics of a cancer usually means that you have a mouse model that you can study and manipulate in the laboratory. And as I said, we don't have a mouse model for neuroendocrine tumors. We don't have a good one. And so we're trying to develop one so that we can expedite the progress of understanding the genes. Then there's an entire field of epigenetics. Epigenetics are all the things that go on in a cell that can influence which gene gets turned on, which genes get turned off, without an actual alteration in the gene itself. So epigenetics is huge, and so we're combining now efforts with experts on genetics and experts on epigenetics. There's a concept of drivers and passengers. So an individual cancer cell might have 100 mutations, it might have 10,000 mutations. But which of those mutations are important? Because cancer cells grow, they divide, they make mistakes, there are a lot of mutations that are just kind of along for the ride. They're not important mutations in terms of how that cell operates, they just kind of happened, and they get propagated. We need to find a way of deciding which of those mutations are important mutations that drive the cell to grow, these are called driver mutations, or which ones are passenger mutations just going along for the ride. So what are cancer genes? And we know this mostly for other cancers. We know that the genes that ordinarily, in normal growth, the genes that dictate growth or promote growth, when they go wrong, when they're inappropriately turned on, we call them oncogenes. Why? Because if normal function is to promote growth, and if they're broken, like a light switch, but broken in the on position, so they're always firing, then the cell's always growing, and that's what a cancer is. So oncogenes are one half of the puzzle. The other half is, well, what about the genes that are intended to turn off growth in a cell? There are genes that we have that slow the growth of the cell. Well, if those get mutated and get broken in the off position, so they're not working anymore, then the cell keeps growing. So you can have a cancer by turning on an oncogene or by turning off a gene that suppresses cell division, and when that happens, that's called a tumor suppressor gene. Turns out that most inherited diseases are due to defects in tumor suppressor genes. So there's this delicate balance in normal physiology, normal growth, where the genes that promote growth and the genes that inhibit growth are in this nice balance so that cells only grow when they're supposed to in the normal tissue. But a cancer tissue is not a normal tissue. They might have oncogenes that are driving its growth, or they might lack the tumor suppressors that are inhibiting the growth. Either way, you end up with inappropriate growth, and you get with more cells. The cells are able to develop the ability to recruit blood vessels to feed them. They're able to invade in other tissues. These are all distinctive processes on the step towards cancer. So here's a famous experiment that was done by one of our professors here, who was working with Dr. Bishop when he was at UCSF and got the Nobel Prize. This is the fat tumor on the left is not from donuts. Fat tumor on the left has a lymphoma, and his belly is full of lymphoma. But this is a mouse that was modeled to turn on an oncogene, and when that oncogene is turned on, they get a lymphoma like that. But this mouse can be manipulated so you can also turn off that oncogene, that single gene. So when that gene is turned off, that lymphoma disappears. That's the same mouse on the right after treatment for this one gene. So that gave us hope that if we could just identify the oncogenes, we might develop some form of genetic engineering or some form of targeted therapy to turn that gene off. And that has been true in a small subset of a number of cancers. Lung cancer maybe most notably, some other rare cancers, the GI stromal tumor, some leukemias, where we've identified the single gene that causes the cancer to grow. We can knock it out, and patients can live a happy life in remission. So that mouse is a proof of concept that strategy can work if we can identify a gene. But not all cancers have an oncogene. So what do we know about the neuroendocrine tumor genome? That's what you're here for. You don't care about the lung cancer genome or the breast or others. We're talking about neuroendocrine here. Well, we first know that there aren't as many mutations in neuroendocrine tumors. This is a plot where it goes left to right, and the tumors on the right are going up. That means they have more mutations per cell. So the ones at the far right are melanoma. Melanoma has a lot of mutations per cell. The ones at the far left are childhood tumors, which have relatively very few mutations. And look where carcinoid sits. If you can't see it, carcinoid is that box on near the left. So not nearly as many mutations as many of the other tumors. And one of the reasons that we're concerned about applying immunotherapy to carcinoid tumors, and one of the reasons we're concerned it might not be as simple as we'd like it to be, is that all of those stars over those tumors on the right are tumors where there's already an FDA approval for an immunotherapy, or the open stars are stars where we're almost certainly going to have an FDA approval within the next 6 months. They're all clustered towards the higher mutated tumors. So you can see that carcinoid's way down the list. So we're going to have to find a way to convince the immune system to react against carcinoid. It may not be as simple as just giving them one of these new drugs. Now, there are some genes, and I'm not going to bore you with a lot of the names. There's one that's called CDKN1B. It's a gene that encodes a protein, and as all genes do, and this protein is called P27, and it's a cyclin-dependent kinase inhibitor. It's a potential tumor suppressor gene, and it can be mutated in about 10% of carcinoid tumors. Now, that means 90% we have no idea, and we're not even absolutely sure that the 10% who have CDKN1B mutations, we're not exactly sure that that's the driving force. So we're still early into this, and a lot of research is going into this now. There's a second group at NIH that has looked at familial carcinoid, And they found in a number of families that had carcinoid that there's an IPMK mutation. So it's an inositol polyphosphate multikinase. Don't worry about the number of syllables. But that might be responsible for some subset of inherited cancers. And there are a number of experiments now going on to try to confirm that, and then just test if that particular gene or that particular protein might have an influence on any in vitro model that we develop, any mouse model. What about pancreatic neuroendocrine tumors? Well, there are a few more mutations in pancreatic neuroendocrine tumors than there are in midgut carcinoids. And the ones that are characteristic of pancreatic neuroendocrine include the DAX, ATRX, TSC2, PTEN, PI3 kinase. This is just a table of the most common mutations that were found in a study that was funded, by the way, by the NETRF that was done at Hopkins. So the Hopkins folks took a group of pancreatic neuroendocrine tumors, did sequencing, came up with these mutations in pancreatic neuroendocrine tumors. So now I'm going to divide that into the chromatin ones and the mTOR ones. And so if you look at DNA, as I said, it's more than what changes occur in the DNA, which is what sequencing is all about. We're literally kind of reading the blueprint of DNA. It turns out that DNA can be genes within DNA can be turned on or turned off based on what proteins are around the genes, and that's the chromatin. So these are chromatin remodeling genes. The MEN1, the DAX, and ATRX can affect how chromatin and other proteins bind to the genes and either hide the genes or help the genes unfold so that they can be expressed. Similarly, the other genes in that pathway were PI3 kinase, PTEN, and TSC2. Those are all genes in the same signaling pathway. They're in the mTOR signaling pathway, and coincidentally, one of the best drugs we have for pancreatic neuroendocrine, and as of last March, also for midgut carcinoid, is everolimus. Everolimus binds to the mTOR drug here, and we're hopeful in that study that you heard about that Emily mentioned from the NCI is perhaps if we select for the PTEN, PI3 kinase, or TSC2 mutations, maybe we might be able to get more bang for our buck by focusing on drugs that hit those mutations. And there's a number of clinical trials. We're about to open a clinical trial with TAC228 for people who've had everolimus where everolimus is no longer working, because this may work on a pathway that renders resistance to everolimus. So we're making small incremental gains in our understanding. Some of these are coming from the genes, but I have to tell you that some of these drugs work even when those are not mutated, so that's not the full story. I'm not going to go into this slide that's too complicated on the left, except to say that poorly differentiated neuroendocrines, which you've heard this morning, are completely different from the well-differentiated neuroendocrines. Not surprisingly, they have different genetic pattern. So the poorly differentiated neuroendocrine tend to have P53, RB1, and RAS mutations, a completely different set of mutations that we typically find in other carcinomas, in adenocarcinomas. Whereas the well-differentiated neuroendocrine have the ATRX, the DAX, the MEN1. So what's on the horizon? Certainly it's been daunting, it's been disappointing that we haven't found one gene that we could target. But we've decided to do, as I mentioned, one of our accelerated grants will go to a comprehensive genetic and epigenetic characterization of small intestinal nets. It can be the most thorough analysis of small intestinal nets that's ever been done. And it's being done, by the way, on tissue specimens from a surgeon at UCSF who's donated that to a group that will be working on that. And in fact, the group is in both Harvard, at the Broad Institute in Harvard, and also in England. So this is really an international effort. Circulating cell-free DNA, I'm not going to get into that at all, except that it's a potential way of doing a liquid biopsy. We get a blood test, we can measure the DNA in it, we can look for mutations, and perhaps we can use that either as a tumor marker that might be better than chromogranin A or some of the other tests that you've heard about, or perhaps we can learn what mutations are going on in real time in a patient and then react to them before we have to see the CT scan. That would be the goal of that kind of research. We're not there yet, but that's one of the avenues we're approaching. An in vitro model, we don't have a good mouse model. We don't have a good model in the Petri dish. So we're making efforts to try to develop an organoid culture as well as offering prizes for anyone who come up with a Petri dish. So if you want $250,000, just go to your garage, start working on the Petri dish. If you come back with a model of a neuroendocrine tumor that grows in a Petri dish, Ron will give you $250,000. And then we're exploring, one of the exciting things is the immune system just needs to recognize something foreign. Now, if you have lots of mutations, you're more likely to have something that looks foreign. But all the immune system needs is one thing to look for in. So if we can find out one foreign antigen in the neuroendocrine tumor and convince the immune system to go after that, we can make some headway in immunotherapy. So one of the avenues is to explore the mutations in neuroendocrine tumors, try to find out which ones might look juicy to T cells, and then see if we can amplify a T cell response to make the tumors responsive. So with that, I want to actually, and by the way, I've asked people many times before, have you ever been to campus? And they say, oh yeah, I've been to football games, or I've been to these things. Campus is beautiful. So if you have time at the end of the meeting, just walk that direction, and you'll be in the old quad. It's absolutely gorgeous. Walk that direction, and you can see the museum and the outdoor sculptures. So I hope you do have a chance to enjoy it. If not this time, the next time we have it. So Pam?